Okay, so we're going to look at one of the big topics of the Lead and Cert biology course, at least one of the big topics from the human physiology section, and it's the function of the nephron. So I said in one of the last videos, and there's a video on the structure of the kidney. One of the structural subunit, or the structural functional unit of the kidney, is called the nephron. And there's millions of nephrons. Each kidney has lots and lots of them. They're very tiny structures. And we're just going to look at the structure of it and also how that relates to its function. So we said before that the function of the kidney is twofold. We said that this is functions of the kidney. Let's just, it's, if you like, functions of the nephron, just generally speaking, before we look at the detail. The two main processes we're going to keep in mind all the way through this story as we go through the nephron. One of the functions is filtration. Now, we'll see that this is often referred to as ultrafiltration because it's filtration that occurs under pressure inside the kidney and the nephron, and we'll see how that occurs and why it occurs. And then once the blood has been filtered, and of course the reason that the blood is being filtered is to remove waste products, mainly urea, but also uric acid, and some other toxins and things that are to be eliminated, removed or excreted from the body. Removed from the blood and end up in what's called the filtrate of the nephron, and ultimately will be eliminated as urine. So the formation of urine involves this filtration process, or ultrafiltration as I said, and it also involves what's called selective reabsorption. And it's interesting, and unless it's sort of brought to your attention, you probably wouldn't necessarily question why we're calling it reabsorption. So after being filtered, the blood, there's lots of things that come through in the filtrate that you don't want to lose from the blood, so they have to be taken back into the blood, and so they're returned to the blood. Now, why we don't call this selective returning, and why we call it selective reabsorption, so this word reabsorption is important. And actually at least one year it came up on the Lead and Cert paper. They asked, why is it reabsorption? And the reason it's reabsorption is because a lot of the materials that are being taken back into the blood after being filtered have been absorbed once before in the body, and that's at the small intestine. So a lot of the stuff that's reabsorbed back into the blood after being filtered in the first place by the nephron, a lot of this stuff is food molecules that have been absorbed once before from the wall of the kidney, the wall of the small intestine. So if you remember the villi, the wall of the small intestine would be involved in absorbing amino acids and glucose and so on, and these molecules are filtered in the nephron, filtered out of the blood, and then reabsorbed back into the blood. So this is why we call it reabsorption, just to bring that to your attention. So okay, let's look at this structure of this unit, or the structural unit of the kidney, which is called the nephron. And what we'll do is we'll draw what's called the Bowman's capsule. And the Bowman's capsule is, if you like, think of it as the funnel. When you did your filtration experiments in first year, if you remember, inside the funnel goes a filter paper, and then you pour your mixture that needs to be filtered, and some parts of the mixture will go through the filter paper and some parts won't. And so what happens is we have blood is brought into this capsule, and of course we're drawing it in 2D here. Please bear in mind that, of course, this is a 3D structure, and it's more like a cup than a U-bend or than a U-shape than it looks like here. But blood is brought in, as we said before, from the renal artery. So if you can imagine blood coming in to the renal artery from the renal artery here, and a branch of the renal artery, which is known as the afferent arteriole. The afferent arteriole brings blood in to what's called the glomerulus. And the glomerulus is, so this glomerulus, not an easy one to pronounce, so it's GLO. I'll just try and do a better job of that. It's the glomerulus, so glomerulus. Now, it's just a network or a bed or whatever, a tangle of capillaries. So this big windy place of capillaries is held inside the Bowman's capsule. And so this is going to be where the filtration happens. You could say that the glomerulus is partly what the site of filtration. You could also say it's the Bowman's capsule. It's 50 of one. It's half of one and what am I trying to say? 6 of one and half a dozen of the other. So blood comes into this glomerulus, and then it exits via what's called the efferent arteriole. So it comes in what we'll call the afferent arteriole. I'll just put a label in here for that. This is the afferent arteriole. Afferent arteriole. You'll notice that the nephron has a lot of not very friendly terms, and this is what we call the efferent arteriole. So blood enters the glomerulus via the afferent arteriole, and it leaves then via the efferent arteriole. Now, one interesting thing about these two arterioles is that the efferent arteriole, so that's this one here, the efferent arteriole is narrower than the afferent arteriole. Now, the result of that, or the effect of this, is that there's a very high blood pressure then in the glomerulus, because if you can imagine it, because there's blood flowing in through the afferent arteriole, that's quite a wide pipe. Think of it as a pipe, liquid flowing through the pipe, so the blood's flowing in there. It then enters these capillaries, which are quite narrow, but the problem is also that because the efferent arteriole is much narrower, then you end up with this pressurized situation, and this pressurized filtration that occurs then we refer to as ultrafiltration. And about 20% of the plasma is going to pass in through this system, into the kidneys, and so you're going to get a filtration event happening then, like I said, at this in this Bowman's capsule or this glomerulus. Just label this Bowman's capsule here. This is the Bowman's capsule. Bowman's capsule. Okay, so just to focus in on this then, the blood pressure forces fluid into the Bowman's capsule. Of course, the filtrate then, what does pass through into the Bowman's capsule is going to be things like water, like salts, amino acids, like I mentioned before, and glucose, which have been absorbed already once before by into the blood at the small intestine, in the wall of the small intestine. So they're taken out of the blood here, at least a certain amount of them. So water, salts, amino acids, glucose, but also waste products like urea and uric acid that we really want to get rid of completely. So they are all filtered and end up in what's a very grand term, what's known as the glomerular filtrate. So what ends up inside the Bowman's capsule then, and what then flows along from inside then in between these, inside this pipe that I'm drawing here, this tube, which is known as it leads into at least what's known as the proximal convoluted tubule. Now what we'll do is we'll just draw this as a couple of twists. In reality, of course, it's much more windy. convoluted is the word. The reason it's called a proximal convoluted tubule is because it's near to the Bowman's capsule. So it's the nearby one, the proximal convoluted tubule. Convoluted just means windy. It's a tubule because it's a small tube. Okay, so what's going to happen here? I'll just finish that off there. What's going to happen here is a certain amount of reabsorption. In fact, to be honest, what happens at the proximal convoluted tubule, I'll just label it here, this windy thing here, the proximal convoluted. What happens in this is a lot of reabsorption. So no sooner has the blood been filtered and you end up with this glomerular filtrate, like I said. This glomerular filtrate, incidentally, is going to pass along in here. So we'll just draw a little arrow there. I don't know if you can see that. Proximal convoluted tubule. By the time it gets there, a lot of the content of the blood is going to be reabsorbed. Now, the reason that this reabsorption happens is because there's a lot of good stuff in the blood that really doesn't want to be lost. We spent a lot of time and energy getting this good stuff into our blood by locating the food and eating the food and digesting the food and then absorbing it. So we really don't want to lose all that valuable amino acid, glucose, and various other food molecules that are in our blood, that we need in our blood. So all of these, all of the glucose, all of the amino acids, they are all reabsorbed in the proximal convoluted tubule. So I'll just put that in there, that we have glucose, we have amino acids. They are all reabsorbed, so that's 100% of that. But also, what's reabsorbed is about 2/3 of the water, and in addition, about 2/3 of the salt that needs to be reabsorbed into the blood. Now, the salt will cost ATP. It will require some energy to get those to be reabsorbed. Water will be reabsorbed by osmosis, of course. So the glucose and the amino acids, that costs as well. That costs the body ATP to get that to be moved back into the blood, because it's against a concentration gradient. Okay, so let's continue on. We've got a lot of reabsorption has already happened. If we follow this tube on, then it goes. This is the very long tube that we saw in the last video, which was just the general structure of the kidney. And I showed you just the position of the nephron. And this loop here, which goes from the cortex down into the medulla. Now I'll draw you a little line in a minute to show you what I'm talking about. But this is what's called the loop of Henle. Now, when you're drawing this, if you're asked to draw this with Lean Cert, I'd really advise you to label both arms of this, or both limbs rather of this. The loop of Henle, then, it's the loop of Henle. And you have two loops. You have two limbs, rather. You have the ascending limb and the descending limb. So, as it's called the descending limb, and we'll look at that first, as the glomerular filtrate then passes down here through the descending limb of the loop of Henle. And this descending limb that the filtrate is flowing down through is permeable to water, but not permeable to salt. So water is reabsorbed as the filtrate flows down through the loop of Henle, through the descending limb. And the reason that it does flow out to such an extent, we're talking about 5 to 10% of the more water is reabsorbed by osmosis. The reason it flows out, of course, by osmosis is because the cortex is, or rather the medulla, so this the lower part, the inside part of the loop of the kidney. Just draw in the layer of the level of the medulla here, the division, if you like, between the medulla and the cortex. We're talking about this sort of layer. So here we have above is the cortex. So I'll put that in there. That's the cortex, and in here we have the medulla. Okay, so the medulla becomes salty. Now, why is the medulla salty? Well, that's the story of the ascending loop. The ascending loop, so that's this bit here. As the filtrate goes up the ascending loop, that ascending loop is impermeable to water. So, by active transport, the salts will be actively pumped out into the cortex, into the medulla. So the medulla becomes increasingly salty, and of course the reason that this salt, these ions, and we're talking about, just to put them in there, we're talking about the sodium ions, we're talking about chloride ions, we're talking about potassium ions. Okay, so all of those ions are actively, and at the expense of ATP, pumped out by active transport into the medulla. So the medulla becomes increasingly salty, and that of course explains why it is that the more water is lost by osmosis here. So so as the filtrate then moves up the ascending limb of the loop, and we'll go back to our journey, it then will encounter another convoluted tubule. So if we draw our tubule again, this one, you might be able to guess the name of this one. This one isn't the proximal convoluted tubule. It's not the nearby one. The proximal one, remember, was over here. After the loop, now by the time this filtrate has reached the proximal convoluted tubule, it's pretty much in its final sort of form. So the distal convoluted tubule is labeled here. That's what this is called. This is the distal convoluted tubule. By the time the filtrate is reached here, it's pretty much in its final form. So the distal convoluted tubule's role then is to fine-tune the concentration of the salt in the blood, or if you like, the contents of the urine. So there's a small amount again of water, just the final amount, and it's variable, but up to 10% of the water then can be reabsorbed here by osmosis. And we'll see later on that's influenced to some extent by a hormone ADH, depending on sort of the state that the hydration levels of the body and the level of water in the blood. So this can reabsorb up to 10% of water, or it can be, and a certain amount of salt is reabsorbed here by active transport. So this is very much the like I said, the fine-tuning sort of role of the nephron. So the last stage then is, once the filtrate has left this distal convoluted tubule, then it enters a collecting duct, and this is a larger tube, but if you like, it's considered like the drain for a lot of different nephrons. So lots of other nephrons. So there's a nephron drained into there, and maybe another one will be draining into here, and so on. So you get these, well, like I said, they're called collecting ducts, and they collect the filtrate from, so collecting, we'll have to do better than that, collecting ducts. Now, by the time the filtrate has got to the collecting duct, bear in mind that we've now reabsorbed everything that's useful from the filtrate. So the collecting duct is going to be it's going to be collecting what we can call at this stage urine. Now, the concentration of the urine, and it will be quite concentrated at this stage, a lot of the water that was in the original glomerular filtrate, if you remember what we talked about back here at the glomerulus level, a lot of that filtrate has been reabsorbed now. So any of the important, the useful things like the glucose and the amino acids, some of the electrolytes, the salts, and a lot of the water has been reabsorbed. To some extent it's been fine-tuned at the distal convoluted tubule, which we talked about here. That's the distal convoluted tubule. But at the collecting duct level, the permeability of the walls of the collecting duct is also influenced by this hormone ADH. And this hormone ADH then will signal the collecting duct to either become permeable, the walls of the collecting duct to either be permeable to water, in which case water can be reabsorbed back into the blood, or not, in which case if water needs to be, if there's plenty of water in the blood then, and water doesn't need to be reabsorbed, then more water, a more watery filtrate or a more watery urine can be produced. Now we'll look at that in a bit more detail later on. But like I said, suffice to say that between the distal convoluted tubule and the collecting duct, they're both very important in regulating the water levels, or the amount of. Right. But ultimately, then the filtrate that goes through here, or what we can now I suppose call urine, consists of mostly waste products. You're talking about urea and uric acid, salts that we don't need, and whatever water we don't need in our body as well. So all of this then, these collecting ducts, various collecting ducts, will drain into what we looked at before, which is the pelvis of the kidney. And each of those, the pelvis then of course will itself drain into the ureter, and the urine will be carried off into the bladder. Now, just to look at this, the end of the story, I suppose, and it involves this antidiuretic hormone. Antidiuretic hormone, also known as ADH for short. ADH is produced in the pituitary gland. And what it does is it's involved in controlling whether the distal convoluted tubule and the collecting duct, whether they reabsorb water or not. So it's produced in the pituitary gland. And the reason it's produced is as a result of a message from the hypothalamus. So receptors in the hypothalamus, I'll just put this little story here in another color. So the story, if you like, the role of ADH is it's for controlling, like I said, it's for controlling whether the DCT, the distal convoluted tubule, and the collecting duct, CD, whether they reabsorb water or not. So the walls of the collecting duct and the walls of the distal convoluted tubule are affected, or can be affected, by the hormone ADH, this antidiuretic hormone. And the effect of the action of ADH is to make the walls or the membranes of these two tubules permeable to water. So when are they produced? Well, receptors in the hypothalamus. So we've got this area in our brain, the hypothalamus, and it has receptors. It's constantly monitoring. One of its jobs is to monitor the water levels of the blood. So it monitors the H2O levels in the blood. And if it finds that there's too little water, the water levels in the blood are too low, in other words, the blood is too concentrated, but there's too little water in the blood, so it's monitoring water levels as opposed to salt levels, so that's important. If it finds that the water levels are too low, if the water level too low, if the H2O level is too low, then what that causes is it causes the hypothalamus to signal the pituitary to produce ADH. So the result of the production of ADH is secreted into the blood by the pituitary gland, and off it goes to the kidneys. And then, like I said, what it does is it increases the permeability of the walls of the DCT and the collecting duct, so the water can be reabsorbed. So the result of that is that the walls of the CD and the DCT permeable, and you get water reabsorbed. And so in that reabsorbed, and in that way then the water level of the blood is increased, and until such time as the hypothalamus is satisfied that the water levels are okay, and then it switches off, sends a signal to the pituitary gland to switch off. So let's just put that story in as well. The other side of this, the flip side I said, is if the water level, just put it in here, if the water level is too high, or high enough, let's put it that way, maybe that's better, is high, then what a signal is sent. Signals pituitary gland to stop producing. To turn off ADH production. And of course the result of that then is that water stops being reabsorbed, no more water reabsorbed. Let's see if we can do better than that. So no more water is running out of space here, as you can see. ADH is off, so H2O is not reabsorbed. And in which case the water levels of the blood are left alone, and because they're perfectly fine, and we eliminate the water that's unnecessary then, and it's because it stays in the filtrate into what's now termed the urine. So the urine then, the final product, like I said, drains from the collecting ducts into the pelvis, and off it goes into the bladder.